one extra minute and we can come back.
Yeah, sure.
Okay, let's go. We can start again.
We can start again. So we will now enter in the second panel discussion on the way on transferring the nanomedicine toward the market. It's a matter of producing the nanomedicine in a factory and not in your lab. And we will discuss about that, the manufacturing in GMP condition. And I will let Frank Pavan to give the introduction talk. Maybe Frank, I'll let you introduce yourself.
Yeah, thank you. Happy to be with you. So basically, I'm the director of V-Nano. V-Nano is a brand new company which has been created about 2 years ago with the main purpose to provide CDMO activities in charge of nanovectorization. For that, we bought a special device which is dedicated to nanovectorization using several technologies, including polymer nanoparticles and also liposomes, and also other type of nanoparticles using one single technology, which is approved for scaling up, because we can go up to 50 kilos nanoparticles in GMP environment. So that's my current job, and I'm a biochemistry engineer as a background, and I've been working in sterile manufacturing for more than 20 years. I've been involved also in my past with a Refine consortium. That's where I knew Ruth, and I'm happy to be with you today, Ruth, to introduce this workshop.
Thank you. So Frank, we'll let you maybe do the introduction and also introduce your speakers.
Yeah, so we will have a nice speakers today. We will have Claudio de Luca, which will follow my first introduction presentation. Then we will have Loredana, Cecchetelli. After that, we will speak about a little bit about other projects. Then we will have Alba Cordoba from Nanomol, and also Fabien Montury in France, who will be joining at the last introduction of the New Deal project. Claudio is the Vice President for Strategic Development in Finceramica. He holds a degree in Biological Sciences and a PhD in Bioengineering. He has been executive in life science for 20 years, in management experience at the international level, impacting corporate performance through different orchestration of high-level projects. He was a top executive in several startup companies and led new business development in mid-sized corporations, served a variety of leadership positions in research, product development, clinical, marketing, and international sales and operations. He has a broad knowledge about biomimetic materials, medical devices, and cell-based therapies, particularly in orthopedic, advanced wound care, and neurosurgery. He also has a long experience with international regulatory approvals, operation and supply management, strategic negotiator of supply, distribution, and licensing agreements. So that's the next step. Loredana Cecchetelli is 30 years experience in pharmaceutical research and development since 1987. She's graduated in chemistry from the Rome University. Since 2006, she has been the Director of Research and Development of the Istituto Biochimico Italiano. Today, her team deals with all the activities related to internal R&D projects and contract development and manufacturing, from laboratory trials to GMP production of investigational medicinal products. She is also responsible of the biotechnology department, where fermentation and purification processes from mammalian cells are developed. Then we will speak to Alba Cordoba. We will let Alba speak about other aspects. Alba Cordoba is a doctor in technology transfer, director of the SME Nanomol technology, where she heads scale up and engineering since 2017. Also managing business development activities. She is chemical engineer, postgraduated in technology transfer and innovation management, and holds a PhD in chemical science and technology. She has 14 years experience in industry academic collaboration for the development of novel micron and nanostructured materials and biomaterials for health application, having participated in more than 25 applied R&D projects, including research and technology transfer contracts with international biotechs, pharmaceutical and chemical companies, also in H2020 funded projects such as Farm for Fabry. In the last years, she has led the successful scale-up of the DOLAS platform, a unique and green drug nanoformulation technology, until pilot plant scale. Currently, she's guiding the CGMP implementation of the platform for the manufacturing of clinical batches of DOLAS-enabled nanomedicines. Then we will have Fabien. Fabien is responsible of the technical platform NanoApp based in CEA Toulouse, France. This lab is dedicated to the scale-up of nanoformulated products. It involves pilot batches manufacturing from bench to few liter scale in clean area. Before joining CEA Tech, Fabien worked during 20 years for scale-up and prototypes manufacturing at the Pierre Fabre Dermocosmetic. As a senior process engineer, he contributed to launch hundreds of dermatological cosmetic market. Sun cares, shampoos, gels, lotions, creams, preservative-free products, and anti-wrinkles fillers. His expertise is about products formulation, chemical engineering, and sterile processes, with a constant concern to industrial endings. He started as process engineer in vaccine industry in purification formulation at Sanofi Pasteur in Boehringer Ingelheim. That's a good introduction of our speakers today. I hope everybody will enjoy our workshop.
Okay. So we let you sharing your screen for your presentation?
Sure. Okay, so I already introduced myself. We'll try to have a 10-minute speech regarding the activities of nanoformulation manufacturing at the GMP scale, improving the ability of the drug products to move from lab scale benchtop manufacturing to a nanoformulated environment where you can have enough quantities to be able to supply the clinical scale trials that you are aiming. And for that, we'll go on a quick overview of the different activities that are used in order to manufacture the nanoformulated material, potentially the market that we are talking about. Then we will go from theory to the nanoformulation, what is usually the main aspects, but we won't be able to cover all the aspects of the manufacturing, but just a global site view of the different top-down and bottom-up processes. What are the technologies that you can use for that? What is the scale-up and scalability processes that you are implementing in order to get to the right scale? We will touch base on QC characterization. This has been also something that has been covered by the preliminary discussions, and we'll introduce our GMP production facility for the clinical to the commercial scale batch. Short overview of the market. Basically, this market is highly dynamic. We can see it also on the ETPN members, and the number is increasing thanks to ETPN dynamics. This market was in 2017 around 21 billion euros with an expansion of around 8%, 10% increase is expected until 2022. We've seen some of the key products coming, for example, on the COVID-19, which are using also nanoparticles. This is the case of Modena, I assume. There is a poor CDMO capacity for sterile manufacturing. Mainly, if you want to manufacture sterile products, it's difficult to find capacities available in the CDMO workspace. And the main products are manufactured in-house. If you are looking at the FDA-approved nanomedicines, most of them are manufactured by the pharmaceutical laboratories themselves. Baseline, the nanosizing perspective. So here is a short diagram, which all the attendees knows very well for sure. The surface exchange area is drastically increasing when you are moving down to the nano size, which is not the case when you are at the micronized level. The expansion of the surface area for the nano size particles is much higher, and that's what we are looking at. This has one key elements which we are looking for: the protection of the active ingredient from degradation or destruction. By this way, we can avoid side effects and we can target the different therapeutic cells that we want to kill if we are looking at targeted therapies. This will increase the efficiency of the therapy, increasing local API concentration, for example, and protect the API from before biodelivery. Two main activities to ensure the nanovectors are manufactured. One which is called the top-down, where you are decreasing the size of the particles by moving them from micro to nano. This is on the left side, and there is another type of manufacturing scenario, which is the bottom-up, where you are dissolving or getting the product or the API dissolved in different phases, and then you are controlling the way it will be either crystallized or introduced in other type of particles during the process itself. In order to get this available, there's a mistake, sorry for that on the slide. There is multiple way to get there. One technology is wet ball milling technology, using the strength of the balls in order to destroy and move the products from top to down, the size of the particles. The other is the high pressure homogenization technology. Then you have a controlled crystallization, I mentioned that already, by using on different phases scenarios. And there is, on the top of that, multiple technologies, which are not exhaustive here on the slide, but you've got ultrasonication, microfluidization, high shear mixing, then also temperature quenching could be also that. And here's some others that you can find, which are the core shell particulate system, the microfluidic that is introduced here. There is also electrodropping. There is also freeze drying technology. We will see that in our workshop. There is other type of technologies like supercritical fluid technologies in order to achieve the ability to get to the nano-sized particle. Once you get the particle, then you need to scale up. It's quite easy, usually. Well, not easy, but achievable. When you get to the product on the lab scale at the bench top, you have completed the way to manufacture the nanoparticles, and you want to introduce that in a clinical program. So then you need to think about, is it a non-sterile environment that you want to implement for your strategy, or a sterile environment? This will have for sure a key critical aspect for the next steps when you want to scale it up. Today I will focus my presentation more on the sterile manufacturing, and that's what we want to be a part of the business, because on the other type there is multiple ways to manufacture. For us, we identified that only 2 technologies are fully GMP compliant for sterile manufacturing, which are the upper drawing, which is the high pressure homogenization, and the second one in the bottom one, which is the wet ball milling. Only those 2 different technologies for us are achieving the ability to be fully cleaned in place, sterilized in place, and fully compliant with the way we need to manufacture and approve or propose to the market a contract manufacturing approach, meaning that you don't have an equipment which is dedicated to one type of particles and one type of active ingredient. So in that perspective, these 2 technologies can be IQ or QPQ'd, approved for cleaning in place, sterilization in place, and could be introduced inside isolators, because we have seen in different workshops that most of the products which are looking for moving to clinical scales are cytotoxics, high potent, or used in anti-cancer treatments. I'm going up to the end of my presentation. So in order to be sure that you have manufactured at larger scale the right nanoparticles, because the scaling up is not always an easy way forward. So you need to also well characterize the technology and the nanoparticles you have manufactured at lab scale, in order for us, when you get to a large scale manufacturing, that you get exactly the same nanoparticles that you want to use for your clinical scale material. That's why you need to take care about all the different aspects of the nanoparticles, the surface treatment, the biological treatment, the chemical activities, and the physical properties of the nanoparticles itself, in order to be well identified, in order to propose to the clinical scale material and to answer to the questions from the regulatory people, the right answers and the good product that has been introduced in clinical phase. The solution that we have implemented on site is beamed here with the special GMP compliant equipment that you see on the screen, which is dedicated for HPH, high pressure homogenization, coming from 2 kilos up to 50 kilos, where we can manufacture and fully introduce any type of particles using several phases. This is where we are moving out. The formulation itself is not the only thing that you are looking for when you are proposing or looking for a CDMO activity. You want to get the formulation up to the clinical therapeutic unit. And
Frank,
that's why...
excuse me, Frank. Do you hear me?
Yeah, I'm done.
Yeah, I have a look on your allocated time. We are over. That's why I want to try to tell
Sure,
you.
that's the last slide.
Thanks.
Okay, and this is what we offer up to the therapeutic units. We can go to the vials or the pre-filled syringes you want to introduce in your clinical scale. That's all for me. Thank you for that. And let me leave the voice now to Claudio. Claudio, which will introduce the other topic of the speech.
Yeah.
Okay, thank you. I think you need to leave me share my screen.
And also a word for other speaker, please try to stick to your allocated time, because we will try to save some time for discussion and Q&A session. Thank you.
Okay. So I hope you can see my screen now.
Yeah.
Okay. Thank you very much, and good afternoon to everybody. Thank you for the introduction of myself that Frank has done. So I've been asked to briefly introduce Finceramica as first slide. So, well, we are 50 people in Finceramica, so a small company, a mix of chemists, biologists, material scientists, bioengineers, medical doctors, I would say. And we have R&D in our DNA, because we are a spin-off of a research institute, the Institute of Science and Technology for Ceramics, which is based here in Faenza, close to Bologna, where we are located. And this we were spun off from this institute 15 years ago, or more or less. So we define ourselves as product developer and working in the nanomedicine and tissue regeneration field. With this sort of business model, I'm not introducing that. And basically, we have been able so far to develop and put on the market 3 line of innovative medical devices, biomimetics. Basically, they are scaffolds, bioceramics-based scaffold, bioceramics and some other biopolymers, for which are helping the body to regenerate damaged tissue, basically. So either in this cranial application for regenerating large cranial defect, or in spine and in trauma surgery, or in joint surface preservation. But lately, in the last 4 years, I would say, we have also started to work on nanoparticles. Well, our product has already nanostructured, so the step was easy to move also to nanoparticles, and specifically to ceramic-based nanoparticles, which are the nanoparticles which are using CUPIDO. CUPIDO is the project that Daniele Catarucci just before has described. Is a EU-founded project where the aim is to put together, to combine together a peptide, an active peptide, some inhalable nanoparticles, which are embedded into respirable microparticles, and to deliver this through the lung directly to the heart, in order to restore the myocardial contractility in heart failure patients. The consortium is composed of 12 partners, and Daniele has already described that, so I'm not gonna go through again. So our role in the project was to scale up. We are the scale-upper in the project, and to scale up from the lab scales to the pilot scales, the production of these microparticles, these calcium phosphate-based microparticles, which are embedded in microparticles, as I said, and to scale them up to the grams, to the 100 grams level, in order to allow for the large animal model to be performed within the project, which is the point where we are now. So now we've been able to produce these batches in the order of grams of these inhalable microparticles embedded in the nanoparticles, and run the large animal model. So here you see the nanoparticle, which are platelet-like nanoparticle, is the size of less than 100 nanometers, and which include the peptide also. We produce them through a co-precipitation methods, and then we spray dry them, we purify them, and then we spray dry them into microparticles in the size of less than 5 microns, which are respirable then through the it can be inhaled and go to the lung and then directly to the heart. So briefly, I was asked to describe what were our success factors. Indeed, I would say that the main things we have been taking care of is to get a close interaction with the R&D scientists and the lab scale development team into the project. So meetings, because it's really fundamental from what I have understood. Not only in this project, but also in other projects in my career, that the interaction with the one that's developed the technology to be the most dense as possible. Without that, we are gonna run a lot of dead experiments and not being able to scale up the things. Of course, I think it's important to agree on specific technology transfer protocols. You don't want to leave anything to the uncertainty. You want to plan before what you really want to transfer and where you want to go. And where you want to go is very important. It was highlighted also in previous workshop. You need to know what kind of product you want to scale up. So you need to have a clear target product profile. You need to have clear from the beginning which are the in-process control that you want to have. And also at the end, what kind of certificate of analysis and the structure and the content of these documents need to be well agreed in the development team and well clear. Obviously, you can modify that during a little bit during the scale up because you will face some difficulties in maybe in sticking to fixed specification. But I think that this route has to be really always be clear, and the endpoint of this where we want to go need to be very clear. And of course, you need reliable and reproducible analytical methods. If you cannot trust what you are measuring, that's the end of the story. So you need to have clear analytical method, and with these small molecules, it's not always easy, as it has been highlighted also in the previous discussion in this workshop. Another success factor I see very important is again, design of experiment. You don't want to, even if at lab scale this was done, you want to do it also at pilot scale in order to confirm the parameters and to really scale up the things in a proper, logical, and rational way. If we think about the limiting factor, at least the limiting factor we had during the development, I would say that obviously, yes, most of the scale up activity, you see results at lab scales and you are never able to reproduce that at pilot scale. This is quite normal. Sometimes it's really frustrating. We had during the development some results which were not reproducible, and this has forced us to go back to the lab scales and to redo something at lab scales and then go back to a new process for the scale up and new parameters. Then, of course, adaptation and optimization of the lab scales process was much longer and more complex than originally envisaged. And this is because, as I said, and everybody has said, those damn nanoparticles are really small. And when you investigate them, they are already changing. So, and this is making the things a little bit more complex. So not always easy to adapt and optimize, because sometimes it's difficult to understand if you have really optimized. And then some other key limiting factors are the costs, because we are working with a peptide which is expensive, and sometimes we have been the necessity to go also to external service for some production steps, and this is very expensive. And when you want to scale up and run, large amount of experiment to confirm statistically the optimization of this scale up, this can be a limit. In our case, there was also a complex logistics because we are producing the nanoparticles here in Italy. Then they are sent to Belgium for the spray drive. Then they are characterized in another part here in Italy and in 2 other labs here in Italy. And then they are sent to Germany, where the animal models are done, and also to UK, where the toxicology was done. So a little bit of complex logistics. Finally, what are the challenges to reach commercial scale-up production? So far we are still at that.
Claudio, I don't want to interrupt you,
yeah.
but it's more than 3 minutes over your time.
Okay, sorry. The commercial, this is further process optimization, GMP validation, cost and timeline. Of course, the layout of equipment is important, and eventually. All the challenges for every therapy is that you want to first demonstrate safety, efficacy, and effectiveness of this therapy before moving ahead. Thank you very much, and sorry for having run longer. Thank you.
Thank you.
Okay.
So Loredana.
Yes. Right.
I think it's up to you.
Yes, but
Claudio has to close first.
Close. Thank you.
I cannot. Apparently, I cannot move anything else.
I did it for you, Claudio. No problem.
Oh, sorry.
Just Loredana, Suresh will maybe send you a message on the chat to inform you about the time. Okay. So please keep an eye on the chat.
I'll try. Good afternoon to all of you, and thank you for inviting me. I'm Loredana Cecchetelli. I'm the Director of Research and Development Group of IBI. IBI, Istituto Biochimico Italiano, is an Italian pharmaceutical company founded in Milan in 1918 and located in Aprilia at about 30 kilometers from Rome. Inside, we are present in retail and hospital market, not only Europe, but also in the United States. Inside our manufacturing site, there are also R&D laboratory, and our expertise is mainly in the development of all kinds of formulation, topical, oral, but mainly injectable product, and also in innovative delivery system. In technology transfer and scale up, lyophilization, analytical development, and in 2015, we received also the authorization for the production of recombinant protein from mammalian cell. We have been working in the last 3 years in a Bismarck project. Bismarck project is as the final aim to development a modular nanopharmaceutical with a method that can be applied in an industrial environment to achieve delivery of therapeutic RNA to the brain for the treatment of neurodegenerative disease. Inside the project, there are 9 different partners from institution, university, and the companies, and University of Utrecht is the coordinator. The IBI role inside the Bismarck project is for the scale-up and GMP manufacturing, and we deal with 2 different kinds of nanoparticles. The first one, solid lipidic nanoparticle, that were developed by Utrecht University on laboratory scale, and biopolymeric envelopment nanocomplex that were developed by University of Santiago de Compostela. During the study of the scaling up and industrialization of the process, we follow all the flow sheet of the manufacturing process, starting from the preparation, For the preparation, the consortium decided to use microfluidic technology, and all the partners use the system of nano-precision system. Regarding the purification, we decided to use tangential flow filtration because this kind of techniques not only is easy to apply in an industrial environment, but it is useful to remove residual of reagent and solvent, but also to change the buffer of the preparation if it is necessary to optimize the final formulation. Then surely the formulation, the choice of the excipient that can be qualified for supplier and for analytical characteristic of the material. And more important, considering that inside the project is for a use of the drug for intravenous or for intranasal administration, was necessary to study the sterilization of the formulation. And for us, it was possible to apply a method by sterilizing filtration because both the nanoparticle have a size below 200 nanometer, that is the medium size of the whole of the filter. As a finished formulation, we decided to use for solid lipidic nanoparticle, we have studied two different kinds of formulation, one liquid and the second one freeze-dried. For biopolymeric nanocomplex, freeze-dried formulation was studied. And for all this kind of formulation, we have followed the stability study until 12 months. And we collected the data demonstrating the good stability of all this formulation. To have successful in the scale up of the process, for us the main important topic was apply a quality by design approach. A quality by design approach that is described in a lot of guidance. I would like to point out mainly a draft guidance for FDA that is drug products, including biological products that contain nanomaterial, because in this guidance are approached particular issues very important in case of nanopharmaceutical products. Limits during the scaling up. Surely during the development there were some problems. The first one was the low availability of API and some kind of excipient because they are very expensive. For that reason, we decided to study the scale up using some surrogate products. And use the real material only in the final tuning of the scaling up and the production of the clinical batch. The second problem was the missing of specific analytical procedure and equipment dedicated for a complete characterization of nanoparticle. In this case, the support of the other partner of the project, like SINTEF and Malware, that are specialized in analytical development, give us the possibility to overcome the problem. Regarding the future perspective for the manufacture at commercial scale, surely for the preparation of nanoparticle, we will develop a design of the GMP plant based on microfluidic techniques. To do that, surely it will be necessary a strict interaction between our engineering team and the supplier of the equipment. Nano precision system is already available, a GMP plant that can produce batch to more than 20 liters per hour. Regarding the finished product instead, IBI is already approved for both liquid, small liquid volume and for freeze-dried product. And we have two filling lines, one dedicated to phase 1 and phase 2 clinical batches, and the second one for phase 3 and the commercial phase scale. Thank you. I hope that I stay in the time, in the foreseen time.
Thank you, Loredana. So the next is Alba.
Hi.
Hi, Alba. The same, keep an eye on the chat. Suraj can send you an alert, okay?
Okay. Can you see my screen now?
Yeah.
Yes. Okay. So thank you in first place for the invitation. It's a pleasure to be here in this interesting workshop. And I will try to summarize which was our approach in the scale up of the Delos platform in order to produce robustly and manufacture an enzyme-loaded nanoformulation for the treatment of Fabry disease. At Nanomol Technologies, we are growing SME. We are located near Barcelona, and we are 14 employees now. And what we provide is technological-based solutions for the development of value-added nanomedicines by using our own and proprietary formulation platform, which is called Delos. And we are experts in particle design. And we also have a specialized contract analysis unit that delivers GMP compliant particle characterization services to more than 40 clients, mainly for the pharma and biotech sector. Here you can see a scheme of how our Delos nanoformulation platform works. We use compressed carbon dioxide as a co-solvent, so it is a single step and easy procedure that it is also green, and that can deliver very stable and very homogeneous nanocarriers, such as colloidal systems, for example, liposomal systems, but also other kind of non-liposomal systems, but with a very, very fine tuning of critical quality attributes, for example, with a very fine control of particle size distribution and polydispersity index. And we can integrate a large variety of active compounds spanning from small molecules up to biologics like peptides and proteins. And we have already shown and demonstrated its scalability up to pilot scale level. And we are now facing the GMP design during the following year. And we can provide either a versatile approach for production, either batch or continuous mode. So we cannot foresee limitation for industrial requirements of the platform. And the framework of the Smart for Fabry project, which Nora Ventosa nicely introduced before, our role here was to provide in first place the scale up and the production of the batches that were required to perform all the preclinical regulatory assays. And in second place, also, we delivered a design including detailed engineering of a prototype pilot manufacturing line that will allow a future GMP production of clinical batches. And as you can see in the image below, the nanoformulation consists in a liposomal nanocarrier that protects the GLA active enzyme, which is the drug substance, and also includes a peptide targeting unit that enhances the intracellular delivery. And the main challenge here that we had to face is that these two substances are complex biological substances that were developed as well and supplied by two of the partners with great effort and success during the project. So here we needed to adapt very carefully up to the microgram level to all the available quantities of these very valuable starting materials. No? So the main key for success, and also also linked for the other speakers, was a very close and careful planification of the required amounts in the timeline since the very beginning of the project, and a huge coordination effort among all the partners involved in all the steps from production, characterization, and testing of both the drug substance and the drug product. Here, I'll show you how we did approach the scale up. We did it in 3 steps. First of all, meanwhile the enzyme was being developed, we set up the scalability of the nanoliposomes by using unloaded formulations. And we were able to reach to the pilot plan scale with reproducible results. And then what we did was to analyze which were the batch size requirements for all the further testing that was needed, for the regulatory testing, and selected the proper scale that allowed to use the materials more efficiently and not to waste a single microgram. So secondly, when we had a preliminary version of the enzyme, we also used a quality by design approach, also employed by Luca and Loredana, in order to define a robust manufacturing procedure and to evaluate a design space that you can see here, that it's an area when we can ensure that the quality of the product that we are manufacturing will be achieved, in terms of reproducible critical quality attributes. And once we had this manufacturing process in place, and we also validated the scale that was going to be used, we finally achieved the productions for the regulatory phase with the final version, the pure enzyme, as we did only have a single opportunity. So because the amounts were the ones that were available. So thanks to all the work that was previously done, we successfully achieved all the quality and the stability that was required to deliver the material for the in vivo testing. And just here, just some pictures for you to show you how we produced these materials. Here, the first step of the Delos procedure, when we end up with a liter of colloidal dispersion, which is further concentrated, and the media is replaced to have a suitable in parenteral administration. Here you can see an example of the vials that we delivered, and now we are together with another partner of the project. We are developing a lyophilization receipt that will allow to have a dry powdered pharmaceutical form. And well, thank you for your attention. And I want to thank all the partners of the project. It has been a very, very collaborative environment. And I agree with the other speakers and in this important point of meeting and speaking a lot. And thank you.
Thank you. So Fabien, it's your turn. So we are not in the scale up of the time for speaking, please. So please, Fabien, again, stick your time. Have a look on the chat sent by Suresh.
Okay, so do you see my screen? It's okay?
Yeah, it's working.
Okay, perfect. So let's move to the big scale. Do you see it entire?
Yeah.
It's okay. It was a little bit delayed. So I will illustrate the large-scale manufacturing and the key factors of scale-up with a case study on which we worked in the New Deal project that you already presented nicely, Fabrice. That is called Lipidots. Fabien Montury. So I'm leader of a platform dedicated to the scale-up located in Toulouse. So what are lipid nanoparticles that are obtained with a dispersion of polyphase into a dispersed phase that is aqueous? And the originality of this technology is that we can calibrate the size of the particles so that we can make application depending on the drug we need to address or depending on the application we want to have. This is, for instance, the application we developed in the past. So, for instance, this technology can be used as a way to transport fluorescent dyes to guide a surgery, for instance. But we can also transport inside some drugs, for instance, antibiotics, or otherwise chemotherapy with chemical molecules. But as well, we can also use this technology as gene therapy because we can also fix at the surface some nucleic acids depending on the formulation. And this has been investigated into the New Deal project. Also, we can fix with a covalent bind some proteins and, for instance, antibodies. So that to offer an active targeting. And we can also considering some coupling in between a solution with antibody and a drug so that we can address drug specifically into an organ we can target. So if we have a discussion about lab bench and large scale, this can be really different for many reasons. And for instance, at lab bench, we can do fast screening, especially with some tools like the one that Loredana presented. We can have some fairly simple operations because we can work out on any operations separate. We have also affordable cost because it is small quantities of materials and quite easy to handle because it is small. It is fairly safe if we work into an isolator. When we move up to the large scale, we fairly see that one of the limits is the technology we use at lab scale is not the same as the large volume. And most of the time, I think it's the case also for the other speakers, we have the obligation to move from one technology to another due to the reduced heat and mass transfer, due to the limits of the technology. Also, one point important is the cost and the time of the trials, because when we move up into the scale, we are facing some long operating procedure. And because we also involve some large quantities of materials, we need to consider safety as a priority, and also that is really limiting the environment has to be adapted in terms of sterility, because we are more prone to contaminations. So for this reason, scale-up can be tricky sometimes. For the change of technology, for the lipidots, on the left column, you can see the flow chart. We were working with ultrasound probe to make the nano, and we had to move up to high pressure homogenizer. As an illustration, also, we were moving with dialysis tubing, and we have to move also to tangential flow filtration. So with this, you can see that what was starting point at lab has to be changed to move up to the industrial scale. And this can take time, and it is really important to consider those aspects to have a successful scale-up. First, identify the key process parameters, then proceed step by step, because we have to be careful and confident before moving to the next stage. Repeat your experiments each time at each scale, and be aware that the quality of the raw materials can vary, especially with biologics. With experience, and during this project, we have seen that depending on the source of the materials, we can have different types of results. So make sure that your materials are really reproducible. Then the methods have to be robust and validated, and this is also really time consuming, but it is the only way to make sure that the particles are good, and then compare the results in between the lab, the pilot, and the large scale. And to finish, two important points, make sure that industrial and regulatory constraints are anticipated at the beginning, because this is really confusing and complex afterward to get back to the lab. And one also very last thing that has been pointed out is build a team and have a fluid communication in between the lab and the laboratories. It's really important. That's it. Thank you for your time and thank you for the invitation.
Thank you. So I don't know, Frank, how do you consider the things? But I will let the time, few minutes for discussion and sharing maybe the good practice or recommendation we may have. And I will let Ruth also see if we have question and try to animate also the Q&A session. So we will take maybe 10 minutes, Ruth, for the question.
Yeah, we only have 15 minutes left for the whole workshop, so we have to be a little bit careful here. But I can see that there was one question, and I thought we could shortly discuss this question, and then maybe I suggest that ETPN looks into, because we will have our webinar series, that we quite soon in the early phase try to have a webinar, a follow-up webinar with discussions and more in-depth for only this topic, because I can see that this topic is something that is really important to discuss. But there is one question, and the question to the panel is that microfluidic approach for manufacturing is interesting, and what is your opinion on using microfluidic approaches or technologies, processes? Anyone from the panel that wants to talk?
Difficult to talk for myself, for example, because I choose not to use it for scaling up. So I will let other panelist members to answer, because I heard that one of them is using it to scale up.
But do you have a reason why you don't use it? Is there a reasoning behind?
The reason behind is the fact that when we discussed with a microfluidizer supplier, it was like a black box when we were asking to have design ability to see what are the chambers, how to be clean, how we can have some more activities regarding the GMP necessity to know exactly how the chambers and the microfluidic system is working. And it was like, no, it's our technology, we are not sharing anything. So that's why we considered that's not something that we would implement furthermore if we have to scale up to a large scale projects where they are claiming that they are multiplicating the number of chambers and not increasing the size of the chambers.
Yeah, I think it depends also on the properties that we want to have with the device. I will explain. Maybe for liposomal formulation, relying on a mixture, we can do a lot of steps in microfluidic devices. But if we need, for example, high shearing processes, as we do for nanostructured lipid carrier, we need to put a lot of energy. Sometimes it's a level of high shearing difficult to reach by using microfluidics.
Yeah. But perhaps I can share also, I think indeed there are downsides also to microfluidic manufacture, in the sense that you're dependent on the architecture that is delivered by the company that sells you the equipment. At the same time, what I hear from Polymoon, for example, Polymoon Therapeutics in Austria, Andreas Wagner, they are using a different kind of mixing system, which is based on chaotic mixing, basically. But they say that the application also already in the research setting of microfluidic mixing helps them or makes their job far more easy, because many of these systems at least rely on mixing two different fluids, and that is already much facilitated by if at the research phase you're using a mixing protocol, albeit on microfluidic, versus chaotic mixing in the research phase compared to the production phase. So it might already have its value there that you, during the research phase, employ a mixing strategy that is reproducible at least across labs.
Yeah, so what you say is it's easier to scale up because you can use the same the same principle the whole way during the scale up.
Yeah, and although many people probably will say that you can never compare, or the unique advantages of microfluidic mixing are lost once you step into chaotic mixing, where two fluid streams collide. In the end, for the quality of the product and for the reproducibility of the product, doesn't seem to matter that much. But if there are microfluidic mixing enthusiasts out there, they might feel it. But
any other comments, or should we maybe? I mean, this is something we really need more experience in, because we don't have enough results to compare all those different processes. But I think it's an interesting discussion. But due to the time, I would say, Fabrice, that we continue, and I will discuss with the expo in ETPN on whether we should have a specific webinar on this topic.
Yeah, that's a good idea, Ruth. Thank you.